Generation of bilirubin or catabolism of heme. What are the end product of heme catabolism? It is the bile pigments. Bilirubin and biliverdin are the bile pigments, and this bilirubin as such it has no function in the body. So, it is excreted through bile. The normal lifespan of an RBC is 120 days. The aged RBC, they are broken down liberating hemoglobin. From hemoglobin, the globin chains are separated. So, in hemoglobin, heme is the prosthetic group and globin it is the protein part. So, from hemoglobin, the protein part is removed and this enters into the amino acid pool of the body. Then the iron it is liberated from heme and it is reutilized. Then the porphyrin ring it is converted into the bile pigments. So, this is taking place in the reticuloendothelial cells of liver, spleen and bone marrow. So, from heme iron is liberated and it is reutilized. So, heme oxygenase, this heme it is degraded primarily by an enzyme known as microsomal heme oxygenase. This enzyme it requires NADPH and cytochrome C. What is the function of this oxygenase enzyme? This oxygenase enzyme it will cleave the alpha methynyl bridge between pyrrole ring 1 and 2. So, that result in the formation of a linear tetrapyrrole which is called as biliverdin. And this methynyl group it is liberated as carbon monoxide. So, as we said, the linear tetrapyrrole which is formed by the action of heme oxygenase is known as biliverdin. This biliverdin it is further reduced to bilirubin by an enzyme biliverdin reductase. Next is the transport of bilirubin to liver. Liver plays a major role in the disposal of bilirubin. The bilirubin which is formed in the reticuloendothelial cells it is insoluble in water. So, this lipophilic bilirubin it is transported to plasma with the help of albumin. Albumin binds to bilirubin in a loose combination, that is one molecule of albumin it can bind 2 molecules of bilirubin. Inside the liver cell, this bilirubin that is it is insoluble in water. This insoluble bilirubin it is made water soluble in liver. So, inside the liver cell the bilirubin it is conjugated with glucuronic acid to make it water soluble. The water soluble conjugated bilirubin it is then excreted into the bile by an active process, and this is the rate limiting step in the catabolism of heme. Then this conjugated bilirubin it reaches the intestine through bile. Then what happens? The intestinal bacteria it will deconjugate this conjugated bilirubin. Now, this free bilirubin it is reduced to a colorless tetrapyrrole called urobilinogen, and this urobilinogen it is further reduced to stercobilinogen. Then enterohepatic circulation, about 20% of the urobilinogen it is reabsorbed from the intestine and it is returned to the liver by portal blood. Then the UBG it is again re-excreted, that is known as the enterohepatic circulation. So, after 120 days RBC is lysed and hemoglobin is released. From hemoglobin the protein part, that is the globin portion it is removed and it enters into the amino acid pool of the body. Heme it is converted to biliverdin by which enzyme? Heme oxygenase enzyme. Then biliverdin reductase convert it into bilirubin. This bilirubin formed in the reticuloendothelial cells it is water insoluble. This is bound with albumin and it is carried to liver. Inside the liver it undergoes conjugation. It is conjugated with glucuronic acid that forms bilirubin digluconate, which is water soluble. Then this water soluble conjugated bilirubin, it is excreted into bile by an active process. Now this conjugated bilirubin, it reaches the intestine through the bile. Now what happens in the intestine? There are intestinal bacteria which will deconjugate the conjugated bilirubin. And some amount of urobilinogen, it is reabsorbed from intestine and it is returned back to the liver, that is known as enterohepatic circulation. Then this urobilinogen and stercobilinogen, both are colorless compound and they are oxidized to colored product known as urobilin or stercobilin. This is by atmospheric oxidation. Both this urobilin and stercobilin, they are present in urine as well as feces. So the normal color of feces is due to this compound, and sometimes black color is seen in constipation. Then if intestinal bacteria is decreased, that can be due to the prolonged administration of antibiotic, bilirubin is not reduced to bilinogens. So there will be a green tinge in feces.